Austin, and first of all, I'd like to thank all the organizers today for giving me the opportunity to speak here. And I'll be presenting a novel QTL fine mapping method that incorporates allele-specific data. So before we begin, what motivates us to study QTLs? So it turns out QTLs can tell us a lot about the genetics of complex traits. And if we look at this figure from a GTEx preprint released a couple weeks ago, we see that QTLs are enriched for GWAS heritability. And if you look especially at the rightmost column over there, QTLs prioritized by fine mapping are especially enriched. But more specifically, where does fine mapping fit into all of this? So the ultimate goal of statistical fine mapping is to identify putative causal variants for downstream experimental validation. Particularly, what we want to do is we want to go from marginal association statistics of each marker to posterior estimates of causality. However, the thing is that existing methods cannot narrow down the causal variance to a small enough set to make validation practical. And to make it practical, we would need orders of magnitude larger QTL studies, which would be very, very costly. But what if there was a second source of signal that we could use to narrow down causal variance? And it turns out allele-specific data can give additional insight on cis regulation. And we develop a novel fine mapping, plasma, that exploits this allele-specific data to confidently fine map QTLs. So, but how can allele specificity inform us about QTL effects? To visualize allele specificity, we imagine a scenario where a QTL has a positive cis regulatory effect on expression. And we see that if an individual is heterozygous for the QTL, as shown in the middle panel, there's an imbalance in a phenotype depending on which variant is on which haplotype. And we could quantify this phenomenon as an allele-specific genotype and phenotype. So we first, is this like a laser pointer? Yeah, I just found out about this. Okay, yeah. We first define allele-specific genotype to complement the conventional QTL genotypes, which we define as the phasing, and it quantifies the directionality of phasing among the heterozygotes. And we additionally identify an allele-specific phenotype, which conveys the differences in the phenotype between the two haplotypes. For example, the imbalance of expression between the two haplotypes. And while the QTL phenotype quantifies the total phenotype, the AS phenotype quantifies the difference in the phenotype between the two haplotypes. So we take these phenotypes and genotypes as inputs for the fine mapping, and we feed them into plasma along with additional sample information, including overdispersion and individual level read coverage. And furthermore, these data is used to infer linkage disequilibrium, which is used in both the QTL and allele-specific portions of the fine mapping method. So plasma is an LD-aware fine mapping method. And as a fine mapping method, plasma outputs two outputs. First, the credible set, which we would think of as the smallest set that has a given probability of containing all causal markers, and also the marginal probabilities of each marker of being causal. So now let's take a brief overlook of how plasma conducts fine mapping mathematically. We first calculate marginal QTL effects, betas, under a standard linear model with genotypes X and phenotypes Y. And we additionally calculate AS effects, phes, under a weighted linear model with genotypes V and phenotypes W, along with weight matrix that accounts for experimental factors, such as coverage and overdispersion. And we then model these QTL and AS effects together under a multivariate normal likelihood. And we use this likelihood to sample and assign probabilities to causal configurations. So now that we know about how plasma models allele-specific data, we look at how fine mapping works in practice. And to do this, we evaluate plasma on simulated loci. So for these simulations, we start with 100 real phase genotypes from 1,000 genomes data. And to simulate a locus, we select a contiguous genomic region, and we randomly choose a causal variant from the region. And we use the real genomic data to simulate an allele-specific phenotype at a read count level. And we conduct this process to create 500 simulated loci with one causal variant each, which we'll use for the, I guess, the upcoming figures. So with these simulated loci, we first evaluate how well a fine mapping method is able to prioritize causal variants. And to do this, we use what we call an inclusion curve. So we rank each marker by posterior probability and see how far down the list we have to go in order to get to the causal variant. And we average results across the 500 loci to get a smooth curve. So the first method we compare plasma to is PhyMap, which is a state-of-the-art conventional QTL fine mapping method. We also compare to what we call ASC meta, which is essentially the only other fine mapping method that incorporates allele-specific data that we know of. And it utilizes a meta-analysis of QTL and allele-specific phenotypes. And we also compare it to what we call RASCAL Plus, which is essentially RASCAL extended to produce probabilistic outputs. And you could think of it as essentially fine mapping with a combined allele-specific and QTL statistic. And lastly, plasma, which is the method we introduced today. And we see that plasma substantially outperforms the existing fine mapping methods. If we draw a horizontal line at say about 0.75, we see that plasma much more reliably prioritizes a causal variant at the top of the rankings. And we furthermore, at the top of it. Yeah, and we furthermore look at how confidently plasma can narrow down causal variants in a probabilistic sense, comparing the 95% credible sets produced by each method. And here are shown the distributions of 95% credible sets across the 500 loci. We see that on average, plasma can reliably rule out a large proportion of non-causal SNPs, with many loci fine mapped to single-digit credible sets. So now that we established plasma's performance, we turn to the fine mapping of real data. So the first data set we look at is a large kidney RNA-seq study with 524 individuals, from which we fine map loci with significant allelic imbalance. So on the left are distributions of credible set sizes as a fraction of the total markers in each locus, and on the right are corresponding proportions of loci that are fine mapped within each size. So for example, between 10 and 50, this segment represents the proportion of loci that are fine mapped between 10 and 50 causal variants. So we see that plasma can fine map a significant proportion of loci to a handful of markers. So here is shown as the proportion of loci that we could fine map down to 10 causal variants. And to an extent that's far greater than what we could achieve with conventional fine mapping methods. And to further examine plasma performance, we emulate increasingly smaller studies by downsampling the kidney RNA-seq data. And the numbers below the graph represent the number of loci fine mapped to 10 causal variants using each method. And these downsample results show that plasma can conduct fine mapping at a meaningful extent, even with 50 samples. And we see that plasma at 50 samples actually outperforms conventional methods at over 500 samples. So after seeing plasma's ability to fine map with limited data, we turn to the fine mapping of chromatin peaks using 24 sample prostate H3 K27 acetyl ChIP-seq studies. So while this is not using expression data, this is still a quantitative molecular phenotype from which we could derive allele-specific data. And keep in mind that this data set is too small to be within reach of typical QTL analyses. And as expected, conventional fine mapping doesn't really do that much. Although we don't really get the same single causal variant resolution that we get with the RNA-seq studies, we see that plasma is nonetheless able to get some low-hanging fruit where other methods can't really do anything at all. So now that we have these fine mapped loci, one might ask, how do we know that these credible sets are actually meaningful, especially with regards to real data where you can't directly validate? But what we could do is we see that if we see whether these prioritized markers are in biologically relevant regions. And in fact, studies have shown that QTLs are enriched for functional genomic regions in general, and such as with the study shown in this figure. So to see how each fine mapping method is picking up functional regions, we look at the enrichment of the kidney RNA-seq credible sets with open chromatin regions. And the x-axis here denotes a posterior probability threshold, with a larger threshold indicating smaller, more confident credible sets but also fewer markers. And the y-axis denotes the enrichment odds ratio with the error bars denoting 95% confidence credible sets calculated by Fisher's exact test. So we see at each threshold, plasma's credible sets are more enriched than those from conventional fine mapping, suggesting that plasma is able to prioritize biologically relevant markers. And keep in mind that these fine mapping methods don't have any of this information, this biological function data known to them a priori. So in conclusion, by integrating allele-specific data, plasma achieves a major improvement in power over existing statistical fine mapping methods. And we apply plasma on a kidney expression study, opening up vastly more loci to experimental validation. We furthermore apply plasma on a prostate chromatin study, showing that small QTL studies are amenable to fine mapping. And lastly, these improvements we've shown are effectively for free, since allele-specific data is available for most quantitative molecular phenotypes. And as a next step, plasma could potentially be improved further by incorporating additional data sources, such as functional enrichment data. But more broadly, plasma offers a blueprint for integrating allele-specific data into QTL analyses. And with such a blueprint, we could substantially improve techniques such as colocalization and TWAS and enhance our ability to decipher the biology behind complex traits. So yeah, I'd like to thank my PI Alexander Gusev at the Dana-Farber Cancer Institute, along with all the individuals who helped with data collection, data processing, and the creation of plasma. So thank you all for listening, and I'll appreciate any questions and comments. Yeah.